Thank you very much for your patience. Okay, so since at least 15 years, we have this opportunity to start to study these human endogenous retroviruses, and these viruses are part of us since. These are evolved during millions of years in all the species, and now these are part of our genome. And how this comes? The human endogenous retroviruses come from exogenous retroviruses that have infected individuals, and when a retrovirus infects a somatic cell, the infection is loses after the individual die. But if the viruses, and this counts from these for these viruses, very rarely, if these viruses infect the cell of the germinal cell that will transfer these infective viruses also in the offspring. These viruses are the ability to have an enzyme that is a reverse transcription enzyme that give the ability to the genome that is a RNA genome to be copied in DNA, in a double-strand DNA, and integrated in a genome. When it's integrated, these viruses live with the cell for all the life of the cell. And if this cell is a germinal cell, the offspring will keep in their genome the viruses. And this is the endogenization of the viruses. And through the evolution, all these viruses multiplicate in the genome by their activity or retrotransposition. And during the evolution, part of these viruses remain, very few parts remain intact and with ability to replicate and reproduce viruses. But most of the fragments, sequence that are present in the genome now function only as part of our genome that is also called the non-coding genome, for example. Indeed, the 50% of human genome is composed of retroelements and genetic elements that have the ability to move through the genome. And among these, the 8% is sequenced from endogenous retroviruses. So you can imagine how much this sequence can have function rules also in the function of the genome, of the cell, of the transcription. Indeed, the presence of these herds induce modification in the chromosomes. And also, just thinking, for example, to cancer, this ability to retrotransposition can induce mutagenesis, and in the end, they have connected to the genomic instability. At the same time, these sequences are now enhancers and promoters of functional gene of our cells, and the long-term repeats that are part of that are present at the beginning and the end of the genome of the retroviruses are dispersed in the genome and can be site for transcription factor to transactivate neighboring genes. And this is a very important rule that we will see later also, and it's important to drive plasticity and stemness. Moreover, they can regulate the transcriptome, and also few sequences still can encode for protein, and this protein can interact with the cellular process, or as we will see, can be also oncogenic protein. In the physiology, since of course they are part of our genome, they have a lot of rules. In the physiology, the reactivation of some of the transcription of these retroviruses come to the production of different kind of double strand, single strand, or couple of RNA, DNA that can trigger that so-called viral mimicry. And the viral mimicry is a mechanism that is linked to the pump sensing. So usually when a cell is infected, the virus replicates in the cell, and the immune system is activated because there is a sensing for the detection of these viruses that are replicating, these genomes and atypical RNA, DNA strand present in the cell. And this interact with the toll-like receptor, and so they can activate the pro-inflammatory cytokines. And of course, at the normal level in physiology, this is a normal response, but of course, that could be detrimental in some condition. They activate, of course, as a retrovirus interferons, and so they can drive the response. However, as I told you, when there is a deregulation, this response, the high expression or transcription of these viruses has been associated of several diseases that are linked to inflammation and autoimmunity. But the other side, the herbs have a rule, fundamental rule in humans, since the syncytin-1 produced by herb W is the protein that allows the fusogenic activity to form the syncytin trophoblast. And furthermore, another syncytin from another herbs can act as a regulator of immunosuppression to help the immunotolerance between the mother and the fetus. And going on, also during the development of the embryos, we have different expression of herbs, and in particular RK in this case, that have a timing and specific expression during the develop, and then it's until the blastocyst is formed, and then is downregulated with the outgrowth. And in these in the embryonic cells has been found also detected viral-like particles and protein from the viruses. So they have a functional rule, fundamental rule for pregnancy. And going on, and this is linked to the ability of herbs to regulate stemness in physiology. And this comes important then when we speak then on cancer, because the deregulation of genes that are normally present during embryogenesis are very important and unusually expressed in cancer and in cancer stem cell. So the long-term repeats of different herbs, especially from the family RH and the RK, are for fundamental site for the most of the transcription factor like OCT4, NANOG, so embryonic transcription factor, and this can regulate the plasticity and the identity of the embryonic stem cell. Of course, going on the physiology, we address the normal levels of these herbs in healthy individuals, just as a reference compared on our patient that we can follow for oncological diseases. And what is interesting also to see that they have different expression according to the age, and this is linked also to the function that they have. So how herbs come from, which is the herbs roots when we speak about physiology, but how it comes to pathology. Actually, herbs deregulation has been detected in different kinds of disease that we call complex disease, in which we have different aspects in which, for example, inflammation and the immune response have, of course, a role. And among these, we studied different diseases. We have experience on autistic spectrum disorders and attention deficit hyperactivity disorders. But since a long time, we study these herbs also in cancer. And in particular, we try to address their role as cofactors in the etiopathogenesis, but also if they can use as marker for the disease or to predict the therapy response. We also, with the collaboration with IRA, we have the opportunity to tell something about in a review in seminal cancer biology. And as you can see, there is a lot of papers reporting expression of herbs in different forms, transcripts, protein, viral particles, as well the presence of antibody against the herbs in the serum of the patients. And as you can see, we have different kinds of cancer in which they have to detect. How the herbs can drive oncogenesis. And this could be related, as I told you already, to the chromosomal rearrangement induced by the integration and retrotransposition. And so this ability to give a genomic instability activity to the cell. Then the long-term repeats can link transcription factor, activate oncogenes downstream, or have, for example, RK family induced the expression of protein like MT9, REC, or the HAMV, that has different activity to induce the signaling that induces proliferation or can induce activity in the cell to drive the migration and invasions. At the other side, some epitopes of these proteins have also the ability to immunosuppress the immune response. And actually, we try to understand how also the environment, but more than environment, that it's really complex, the modification of the environment and the susceptibility of Earth to different stimuli to induce the stemness of the cell. Of course, you all know that in cancer, there is actually a complex situation in which the cell under a specific stimuli can modify his morphology, can go in transition from epithelial to mesenchymal cell, and the environment that is called complex can trigger the modification in the cell that drive these features. So our question is if hertz can have a role in this process. At the same time, if we can target hertz to inhibit the activity. And our study comes from the points that different, has been proven that different different proteins, cytokines, vitamins, hormones, and infections of microorganisms indeed can transactivate the expression of the endogenous retroviruses in the cell. The expression can modulate then the gene expression of the cell, the hertz can transactivate, induce the production of the oncogenic protein. They can also, in rare condition, also reproduce viral particles. And this trigger modification in the cell and induction of the immune response. So in our previous work, the first work that in which we, the first study that we did on hertz in tumor was on melanoma, and we isolated a metastatic melanoma cell established metastatic melanoma cell line from a patient, and that is called TVUM-A12. And this cell line was peculiarly plastic and was able to modify the morphological characteristic upon modification of the microenvironment. And we saw that the deprivation of serum arriving passing from 10% of FCS to 1% to FCS modify the growth of the cell from an adherent way to in suspension way of growth, and and the cells start to produce this kind of like clones, we call it clones, that that start to grow very fastly and constantly. And during this modification, we found, as you can see from the transcription, the expression of RK transcription activity was quite higher induced during the exposure to the very low presence of serum, and then stabilize but higher referred to the control when the lines of clones is set up. We found that this, just to come back again in this, we found that these clones has a higher cell growth rate, was able to form colonies. They was able to immune escape response because they are completely lost all the markers of the expression at the extracellular level, at the membranes level, and was able to grow in mice. And peculiarly, we found that after this activation inside the clones, we were able to find the presence of viral particles, presumably referred to RK. And this cell line was, as I told you, very plastic, and we changed the way to to test, we used a specific stem cell serum-free media that is usually used, that's called ex vivo, and it's usually used for hematopoietic cell. This media was able to modify very peculiarly this line, again in the grow in suspension, but with different characteristics. They start to produce a lot of melanin, and they, as you see, they are like dark grades. We have called these grades, and very nicely, this cell were able, by adding again serum in this media, to come back to the former phenotype. And this model was very interesting because allow us to monitor the changing during this evolution. And what we saw that actually during the exposure in ex vivo and the formation of these grades, we had an induction of RK, and this was downregulated again when the cell came back to the FBS condition, the RPMI FBS condition, or ex vivo plus serum. And what is interesting to see that the more differentiated the wild type condition is associated to a normal expression of antigens of the melanoma, the high expression of MSH1, so potentially to present the antigen to the immune system, and low expression of the stem cell marker CD133, nestin, or CT4 and nano. During the exposure to ex vivo, together with the activation of RK, we found a downregulation of the antigen of the MSH1, and so this could relate to an immune escape activity, together with an increase of CD133, nestin, or CT4, and so increase of embryonic genes. What we found that this cell line was, of course, peculiarly endowed with these stemness features, but also in other cell line, we found that an association among the induction in condition of microenvironment change with stress condition with serum deprivation, induction of RK, together with the induction of CD133 expression, and the cell line that has lower reaction to this modification and lower activation of RK, like WM266 or A375, are also express also less cell expressing CD133. So the cell line that has a higher content of stem cell, let's say cancer stem cell, referring to melanoma cell expressing CD133 and expressing embryonic genes, the cell lines that are lower expression has also lower K and the other way around. And then we demonstrate actually that by interference of RK, we demonstrate that the induction and the production of these grades was affected with induction also of deaths, and we could also evaluate that the inhibition of RK abrogate also the expression of CD133, and so the state to that RK activation as a main rule to sustain the develop and the presence of CD133 cancer stem cell in melanoma. Actually, this cell line overexpress FK, CD133 under stress condition, and this was demonstrated to be associated to OCT4 expression. Sorry, here I have to tell that we sorted the CD133 positive cell from this line, and we show the difference between the parental and the isolated CD133, and this cancer stem cell CD133 positive were able to highly induce FK during stress condition, together with the 133, together with the induction of CT4. And there were able to have a higher self-renewal ability compared to the parental. And also they were able to make more migration, more invasion under this condition. So the microenvironmental change induce FK and induce the develop of this cancer stem cell 133. So the issues now is can we target also ERTs to affect this this phenomenon, and indeed also of course with interference we were able to demonstrate that we can stop the cell, but we use non-nucleoside reverse transcriptase inhibitors, or let's say that the drugs that usually are used for HIV, because HIV is a retrovirus, they produce a reverse transcriptases, and this drug, not all drugs for HIV, but part of these, we test different drugs, are able to also inhibit the expression of ERTs, so of the endogenous retroviruses, because the reverse transcriptases of these viruses, it's has a different sequence that and the protein and the amino acid sequence that are highly conserved. And this the use of these antiretrovirals comes to the induction of cell apoptosis of CD133 melanoma cell. And further, we try to test these drugs in melanoma, but also in other kind of tumors. And so using the models of ex vivo and the experience of TVM-A12, we test the response to the microenvironmental change also in FG2 that are hepatocarcinoma cell, and 5649 that are lung cancer cell. And what is possible to see that the stem cell medium ex vivo is able to induce this sphere-like cells formation in all the three cell line. And again, if you can see here the transcription activity of FK, but also of another RH, is induced during the exposure to the microenvironment, and again this was associated to the induction of CT4 here nano, and but less not SOC2. What is interesting also that TVM12 and HepG2 response to this stimuli. But for example, the lung cancer cell 549 have different behavior, and only one kind of ERTs is activated, and for example, SOX2 is activated and not the others. And if we see the expression of the embryonic genes with these hertz, there is a very high and significant correlation of the expression of hertz with embryonic genes expression. And what we can see if we treat this cell line during this process with the antiretroviral drugs. When we expose the cell line to the drugs, in this case are AZT, zidovudina, and the FV is efavirenz. In a normal condition, in standard medium RPMI 10%, the drugs does not affect the cell, rather can little bit differentiate the cell or reduce a little bit the growth, but they don't induce apoptosis, for example. And this, you have to remind that in this condition, the expression of hertz as a standard level that's not induced, is not modified. But in ex vivo, when we induce the hertz, if we during the induction had the antiretroviral drugs, we can affect the formation of the grades of the sphere-like cell in different manner upon the different kind of drugs and upon different dose of the drugs. And what we found actually that we were able during if you see during RPMI on the left side, in the culture with RPMI and serum, there is not a significant change in the expression of hertz and embryonic transcription factor upon treatments, but the significant change is found when we export the cell to ex vivo, when the hertz and the embryonic genes are induced, but the drugs are able to reduce the expression of these genes. And this was happened also on the cell line hepatocellular carcinoma, and also, but different for the genes that are involved for FH and SOX, we have a downregulation also on the lung cancer cell. And we test a colony, clonogenic ability of this cell line in presence also of the drugs. And we found an inhibition of clonogenic activity by the antiretroviral drugs, inhibition of cell growth, and also the induction of apoptosis, but mainly in the cell line that less induced hertz. And why this? We found that the drugs can affect the growth and the aggressiveness of this cell when hertz is expressed, but the induction of apoptosis is higher on those cells in which they are a little bit less induced, because the hertz expression referred a resistance and higher aggressiveness of this cell. And more recently, we also study hertz in leukemia with the collaboration with our hospital, University Hospital of Vergata, and we was aware that was already found, there was already studies on the potential role of hertz also in lymphoma and leukemia, that this can trigger in this cell line signaling important for cancer sustain. And more recently, 2 articles on Nature counts of the important rule of Earth, let's say because it was found transcriptional activated in leukemia cell, and also potentially related to the T cell response. We had 2 funding with fellowship Gilead, sponsored in Italy, and with our colleagues of hematologic unit, we investigate the expression of endogenous retroviruses, again here for chronic lymphatic leukemia, again with the aim also to try to understand, yes, if it's a marker of the disease, if it's important, can be used to monitor patient and to evaluate the response to therapy. And the chronic lymphocytic leukemia is peculiar because it's is a lymphoproliferative disorder of clonal proliferation of B cell, and the incident it's per 1,000 people per year, and it's characterized mainly in aging, and the peculiarity is the clinical heterogeneity. There are a lot of prognostic factors, and the genomic instability in the CLL is really important. Indeed, several mutations are associated to this disease and are a prognostic factor for the evolution. Usually the treatment it's also very heterogenic because usually it's wait and watch because several times there is a chronic condition that can be also not treated, or there is the opportunity to use chemotherapy or more recently biological therapy, and sometimes the transplantation. So the genetic instability, the characteristic of this leukemia comes to the possibility of Earth to have a role in this. So we have studied 50 patients and we had a group of control of 39 healthy control, and we evaluate different aspects. And in the blood of this cell of these patients, we found that different family of Earths like RH, K, and W was significantly high expressed in the patient compared to healthy donors, and correlating the expression of Earths between each other, and we plotting the population, we were able to discriminate the patients using Hertz as markers. And we found a very strong correlation between the expression of Hertz. And what we found also, and this the investigation of the expression of embryonic transcription factor and stem cell marker like CD133 is not very common in this disease. And we found that these markers are overexpressed in these patients compared to healthy control. And if we correlate the expression of embryonic gene with the expression of Hertz, we found a very strong correlation that allow us to very clearly discriminate the patient from healthy donors. And moreover, we correlated the HERK expression to the unfavorable prognostic factor, beta-2 microglobulin. This is an unfavorable prognostic factor at the beginning of the disease. And those patients that has higher HERV expression was the ones that had this unfavorable prognostic factor at their diagnosis of the disease. So the question is case also could be a prognostic factor, new prognostic factor for these diseases. And another interesting results was the fact that upon different kind of therapy, so the difference between the use of chemotherapy or biological drugs change or let's say induce a distribution of the patients in different category if we consider the expression of HERTZ. HERTZ-KH seems not to be affected or not related to different treatments, but RK and W are different expressed in those patients that receive chemotherapy compared to the ones that receive biological drugs. And this can, this is actually is interesting because this gives us the opportunity to try to understand if HERTZ can be used in, of course, in different, as a marker in different therapies, or if different therapy, for example, selects some kind of cell that express higher amount of HERTZ, and maybe this cell could be also a selection of more aggressive that can have a role in the maintenance of the disease. So these are all questions that we are trying to address at the moment. And again, if we see the expression of the embryonic gene factor, the different therapies actually, again, can affect in different way the expression of this embryonic factor. And so we are trying to correlate all this data, and we are going on to monitor the patient to try to find out which is the, the situation. And in vitro, we treat the cell extracted from this, isolated from the patient and cultured in vitro, also with the antiretroviral drugs. What we found that the expression of RK and RH can be downregulated in this cell only in some kind of patient. For example, peculiarly in the patient that were treated with biological drugs. And these patients are the one that has higher expression of RK and H. And we also studied the apoptosis in vitro induced by this this treatment, and we found that the higher concentration of efavirenz, we were able also to induce apoptosis of this cell. And so this means that maybe HERTZ can be also a new target to inhibit this disease. So for CLL, we correlated again, also in this disease, the HERTZ to pluripotency characteristic of the cell and the expression of the transcription factors, and the correlation with the worst important negative markers of the disease. And so the question is if we can act inhibiting HERTs, or also we are thinking about the possibility of immunotherapy. So to conclude, of course, we can act to inhibit HERTs in cancer with different tools. So we are focused on drugs like antiretroviral drugs, because there is a lot of experience already for the use in humans, and we know about the toxicity, but we are also think about immunotherapy. And what is important, it's understand when use the drugs, in which condition, which kind of patients, and of course, now also with collaboration with IRA, we are trying to study the association of this drug also to standard therapy other than chemotherapy, as we show now, also with for example, radiotherapy. And thank you to everybody for this to stay with us today.
Thank you very much, Claudia, for excellent presentation and lecture. And you have already the question in chat. You can read it, and all other seminar participants are welcome to ask Claudia.
Okay. Yes. Okay. I can read the. Okay, if you like, I can read the question. I read yes. The question for everybody from Marina Pukinskaya. Lymphomas are known to often develop in HIV patients. Do you check the HERTs in HIV patients, or do you have any ideas on how they can be related? Okay, very nice, interesting question, because because we are microbiologists, we have the opportunity, of course, to follow also the infectious, the disease linked to the infection. And we have also a funding on the study of HIV on HERTs. On HERTs in HIV patient, and actually there is some literature about this. Is quite new. And it's because they are both retroviruses. Indeed, the reactivation of HERTs during the HIV infection, they regulate also the infection of HIV. In some cases, it's written that can facilitate, in some cases can inhibits also the action of HIV. So what is studied now, if the reactivation of HERTs could be used to monitor for example, the reactivation of HIV, because not at the moment the therapy can stabilize the expression of HIV. So when there is a reactivation, HERTs, the issue is, can we use HERV to monitor the reactivation, because it could be that is a marker of reactivation. So yes, the response is yes, but it's very, very recent studies. There are recent studies on this, so this is very new.
Thank you very much, Claudia. Any other questions? Please use your
okay.
Yeah,
yes.
Additional, yeah.
Yes, it's Cristina Macalli. Thank you. Congratulations for the presentation. Do you know if HER can lead to the expression of immunogenic peptides presented by HLA molecules? Do we allow to consider them as a target molecule of the immune response? Okay, yes. Yes, actually, for example, in the recent paper on chronic lymphocytic leukemia, they found that, for example, the patients that have T cell reactive to FK protein have a different prognosis compared to the one that they don't have. So it is true that they are expressed on this cell. They can be present and they can be used as immunotherapy target potentially. If okay.
Yeah, Berry has.
Thank you for the great talk. Berry,
Thank you.
ciao
Ciao,
Berry.
ciao.
Ciao. Thank you for the great talk. Learned a lot. Sorry for being late. Could you help to understand the cancer stem cell marker expression in PBNC? You mean, could you help to understand the cancer cell marker expression PBNC? You mean the physiological, or you mean the cancer stem cell that they are present in the circle?
Yeah, I mean for the cancer stem cell markers, for example, OX4 and CD133. So why did you check this cancer stem cell marks on the immune cells, on the PBMC cells? So I was a little confusing here.
We in the chronic lymphoma leukemia we checked on the peripheral blood cell because for the kind of the tumors. So usually the only markers that you can see in the PBNC could be CD133, but it's very low in the normal condition. I don't know. I'm not sure if I understand your question.
Yeah, because my question is that these markers usually we check in the tumor cells, right? On tumor cells.
Yes.
So here we check on the immune cells. So I was maybe want to have some
confusing.
Yeah, confusing.
Sorry.
Yeah.
Now, in PBNC, we have found we check the expression only in the CLL because it's a hematological disease. Usually, all the data that I have shown are on the tissue of or cell line.
Yeah, because this is
of tumors. Yes. No, in PBNC, we only check this in CLL. Because
okay, sorry, I forgot this is the
no.
not a solid tumor. Yeah, sorry.
And of course, we as experience we use the expression on PBNC of healthy patient to see the normal expression peripheral. We use as a marker or normal expression of HER2 in physiological condition. But in that condition, if we compare the expression of embryonic genes is not present, and these are our control for CLL, that is a hematological disease. Okay. So the embryonic genes, cancer stem cell genes, associated genes are used only in association with tumors. For our experience, we have this difference always.
Yeah, and I also have additional question which I haven't typed in. Is that because you work on this endogenous virus? So because due to the COVID time, I think maybe there are also some study concerning to check the correlation of this human endogenous retrovirus with the coronavirus patient, right? So for example, for in the future, would
yes,
also
no,
be to check
it's already we have already a preprint in January on this topic, and we are under review for the final reply, and it's already preprint. So yes, the SARS-CoV-2 infection and COVID patients have a higher expression of a specific curve, a specific protein of HER2, and we recently published as a preprint, and we are under review for the final publication. So because we know already, as I told you, that the exogenous infection can reactivate the HERTs in the host, and we associated this activation to the detrimental condition of COVID patient, and there is a correlation with the worst condition.
Okay.
So yeah, they could have a role in the pathogenesis.
Yeah, because I think I also read the news to see like one patient who was have cancer also in a very advanced cases, but he get infected with coronavirus, then the cancer was somehow, the tumors be controlled. So therefore I thought, I'm not sure, you know, so maybe coronavirus somehow also have an impact on cancer stem cells.
I think that the infection with SARS is acute and it's long, a very short time. This experience actually is the number is really few to say that there is some kind of even because the condition for COVID patient is so detrimental that to see something on an evolution of the cancer at the time is nothing.
Yeah, so I would say your talk is really open my horizon because concerning of the cancer microenvironment, you know, so we didn't actually, before for me, I didn't think too much about the virus thing. So I think with your setup, you know, with this, it's really to show maybe in the future, we could also give more focus concerning of the virus environment and as a, also considered as a niche, right, for the cancer stem cells. Yeah.
Yeah, yes. Actually, it's quite complex. It's more we go on, more we know, but more complex is the model.
Yeah, thank you very
much. Of course, we have a lot of new insight to go on to investigate.
Thank you so much.
You're welcome.
Claudia, you have an additional question from Dr. Olat.
Oh, yes, that nice presentation. Thank you. I have the following question. Have you tried to establish any ex vivo based xenograph and treat the mice with antiretroviral drugs in vivo? Does the tumor shrink and produce less metastasis? Okay, so yes, there are data on this on the previously with the normal melanoma cell expressing retro elements, different kind of retro elements, and the antiretroviral drugs in vivo reduce the tumors and the metastasis. Actually, the CD133 isolated can also can give some metastasis, but the higher aggressive are the clones, the one from the first paper that lose all the markers of just let's speaking of immune escape, and they make very big big tumors, and so we have some experience on that, but now we want to go further on the patient. So we will now we don't work anymore with the mice for problem of facility, but also because we want to go further on patient. I hope that the answer was good enough. Sorry, there is written Chiara Vezzani because I'm using the PC on my daughter because of my. No, I'm checking now that the name is the name of my daughter. Because I'm sorry today for this problem of the computer. I have to check it because this is really a problem. Ira, I cannot hear you.
There is an additional question to you.
Okay.
In chat.
Mahmoud, thank you for the great call. Have you tried the combination ZT with chemotherapy agents on cell survival? Okay, we are working on that. There is some already published paper that study the combination could improve But at the moment we are using other inhibitors also in collaboration with IRA that are using during radiotherapy or their rule in radiotherapy. And we have results that the combination increase the susceptibility of the cell, and we have reduction of chronogenic activity and induction of cell death. So the combination is promising.
Okay, are there any further questions? If not, Claudia, thank you very much again for your excellent lecture, and I hope that we will meet in future as well. And you will give us a talk again about new data in collaboration with us, and we could also discuss our interesting data. I have already some results, and I'm wondering whether our your idea would be confirmed during the experiments, and I will invite you again to participate in our seminars, and your colleagues could also participate. And since Claudia and I are the editors in the new journal, Advances in Cancer Biology, Metastasis, we are inviting you to contribute to this new journal. And please submit your papers, and please contribute to this journal. And you can also suggest your special issues and invite the contributors to submit their papers. Thank you very much again, and thanks for all the seminar participants from different countries around the world. I'm very happy that you are interested in our seminars, and I hope to meet you in future, in 2 weeks actually, and you will get additional information about our further seminars. I'm working on that. Thank you very much again.
Thank you to all.
Thank you.
Bye.
Bye.
Bye.